All right, we'll give, if you're logging on now, welcome. This is the Challenges in Pediatric Clinical Neuroscience Research webinar series, and we are going to be getting started in a few minutes. We're seeing our number of participants is going up, and so I want to wait till that plateaus a little bit before we get started. And I'll introduce some housekeeping items. My kid's coming home. I'm hoping that the dogs are not going to be barking, but you never know. I'm going to mute.
I think dogs barking and children in the background are the perfect conversational foil for challenges in pediatric neuroimaging.
Good.
We tend not to have dogs in the scanner or at EEG, but I have to say that children are surprising in their always surprising in their participation.
Yep, absolutely. They can be a big help. Well, why don't we go ahead and get started? Numbers are still going up, but we'll go ahead and get this webinar started. So I'd like to welcome everyone to today's webinar in this series, which is Challenges in Pediatric Neuroscience Research. My name is Adam Hartman. I'm a program director in the Division of Clinical Research at the National Institute of Neurological Disorders and Stroke, which is one of the institutes at the United States National Institutes of Health. I have a number of pediatric clinical trials and other human subject studies in my portfolio, and I'm also a practicing child neurologist and epileptologist. We created the series to help investigators overcome some of the challenges that they might face in pediatric neuroscience areas, including neurology, neurosurgery, psychiatry, psychology, neurodevelopmental disabilities, physiatry, critical care, emergency medicine, pathology, radiology, and pharmacology, just to name a few, and obviously there are a lot of others. The ultimate goal is to improve the quality of care for our patients, but the more proximal goal is to enhance research that will facilitate improvements in practice. Our webinars are targeted to trainees, senior investigators, and even to those who are not currently involved in research, making the pie bigger, so to speak. Today, we are hosting a panel of experts who will be describing and discussing what they see as major challenges in pediatric neuroscience research. As you will see, the discussion will address the topic at many different levels, all in a very thoughtful way. If we could have the next slide, please. So just a few technical notes about the webinar. The session is being recorded and will be transcribed and archived on the NINDS website. Details about where to find the archived version will be posted on social media and professional society listservs and newsletters. There will be a question and answer segment at the end. To ask a question of our presenters, you can either click on the ellipsis near the bottom of your screen and click on Q&A, or open the participant window and on the bottom right, find the Q&A area. To repeat, if you have questions for our presenters, please either click on the ellipsis near the bottom of your screen and click on Q&A, or open the participant window and on the bottom right, find the Q&A area. I will be moderating the question period, and I wanted to offer special thanks to Dr. Adisa Salou from NINDS for all the technical support and coordination of the webinar, and the NINDS Office of the Scientific Liaison for all of its support and the publicity. So let's get started. We're going to start with the presentation by Dr. Bhuma Avarnathan. Dr. Avarnathan is an assistant professor in the Department of Neurology at the Washington University School of Medicine in St. Louis, Missouri. She is a pediatric movement disorders physician and neuroscientist specializing in dystonia and cerebral palsy.
Thanks, Adam. I'm looking forward to sharing my talk with you all. While I'm waiting for the screen to pop up, I think it's really great that we're going to be talking about this from all different levels of training. I represent the early stage investigator of the bunch, so I'm excited to see what you guys think about what I think the challenges are in pediatric neuroscience. When Adam approached me with this topic, I thought right away about the first thing, how we need to combine splitting and lumping. And then over time, I thought about the other two topics, which I'll get a little bit into as we move forward. So as you know, children that we see in pediatric neurology clinic, kids with neurodevelopmental disorders, are all along a spectrum, and that word carries a lot of weight for a lot of reasons when we talk about neurodevelopment, but it's true across all of the neurosciences. And people fall along different areas of that spectrum. And one of the exciting new advances in translational neuroscience is this idea of identifying a specific treatment, an N of 1 treatment for a specific person, and looking at each person individually and thinking, how can I understand your pathophysiology, your specific, the specific genetic mutation that confers the neurodevelopmental disorder that you might be dealing with on a day-to-day basis, and how do I address that? And if you think about that all across the board, you know, we have thousands to millions of people affected by neurodevelopmental disorders. And though the concept of N of 1 trials is really exciting and often the only option available for people with very rare diseases, the idea of cohorting these people or lumping them and research focused on that can also be really valuable. So if you look at all of these people individually, even though they are on a spectrum and even though they're all unique, we know they can all be grouped together in some ways. And maybe the symptoms they share may respond to a treatment that is not necessarily just specific for them, but can address that treatment if studied in a really intelligently cohorted population when studying that treatment and when studying that pathology. So although N of 1 trials are exciting and should be pursued, I would argue that in addition to splitting, we should also be focusing on lumping and funding to focus on intelligent cohorting of clinical populations and intelligently studying a phenomenology as opposed to a specific etiology should also be gaining traction. The next topic, linear and transitioning a linear approach to science to a more simultaneous approach to science. When we think about translational research, I think we think about it in a stepwise fashion typically. So you have a clinical question, you want to develop an animal model for that clinical question. You validate the animal model in some way, you use the animal model to study pathophysiology, that's step 2. Then after studying the pathophysiology, understanding the mechanism, you think about drug development or screening potential drugs after you've identified potential drug targets. And then after that, years and years long process, you may have a few candidate drugs, and then you focus on implementation after the long process of clinical trials and seeing if that drug actually has traction in the real world. And all of these steps are often done by different groups of people. And though there is a lot of collaboration between these groups of people, they often do think about the questions at different times. So instead of thinking about these things in a linear way, is there a way to address all of these aspects of a problem at the same time? And can that be done in the pediatric neuroscience or in clinical neurosciences? And that might seem a little crazy. I mean, how can you implement a drug that you haven't developed about a disease you don't quite yet understand? And as an example of how I might approach this, Adam mentioned at the top that my primary clinical and research interest is dystonia and cerebral palsy. So if you think about how you might approach drug development and screening and pathophysiology right now, let's say we don't have a clear-cut model of dystonia and cerebral palsy, which is one of my passions and something I'm actively working on. Let's say you do the intelligent cohorting we talked about in the first step, and you think about people with comparable etiologies, shared symptomatologies, shared phenotypes, and you look at the comparative efficacy of the meds that are currently in existence. Why do these meds work for this population? Which meds work for this population? Why might that be? And then with that intelligently cohorted grouping of people, you think about what sort of circuit dysfunction do they share? And there are a lot of new imaging and functional imaging techniques that allow us to drill down into kind of a common denominator lesion localization type way of thinking about circuit dysfunction. What are the patterns of brain injury? What are the patterns of brain function that people with a certain type of phenotype share? And both of those research projects can occur in parallel, but it requires those people to work together with similar cohorts of people with similar participants. So having those things happen at the same time in the same research group can be really valuable. And implementation is a process that I think can really start happening at the outset of asking clinical research or translational research questions. Because if we think about the things that we need to study, the questions that may be most pressing to answer, the community has a lot of opinions and has a lot of knowledge about what the issues about their neurodevelopmental disorder affect them the most. For example, for people that do have dystonia and cerebral palsy, pain is a key component of what they experience. So maybe an animal model of that should really focus on having a real pain readout when you're thinking about it modeling the actual disease process. Another thing that the community is really focused on is identifying dystonia in the first place. And we know this through a variety of research projects we've done with the community. So another thing you could focus on with the animal model is, instead of qualitatively characterizing dystonia, which is what has been done in several animal models of dystonia thus far, you could focus on creating an animal model that lends itself to quantitative diagnosis and quantitative characterization of dystonia. But maybe that question wouldn't have arisen without clear assessment of what the community goals were up front, what the community concerns were up front, and what the barriers were to getting the community involved in some of this research up front. So addressing all of these issues at the same time can push research in all of these areas forward. And these people who may have been working independently, if they're forced to share the same funding stream for research that funds all of these issues simultaneously, big multi-PI grants that would like to see a linear approach transitioned into a simultaneous approach, that could make the launching process of projects like this much more efficient and more beneficial to everyone. So the last aspect, community and leveraging community. So I've already mentioned the value of asking the community what questions are most important for them upfront. And the way community has been involved in research so far, and we are as clinicians and researchers becoming more aware of the immense value of involving the community. The community has been involved in fundraising and advocacy efforts, in sharing their stories. What do they think, what has been their personal experience with the disorders that we're all studying. And they've also been involved as research participants, so we've studied them. But what many are realizing now, and what we're working on in the cerebral palsy space, specifically with organizations like the Cerebral Palsy Research Network, is that the community can be an active participant in setting the research agenda, asking them what issues are most important to you, what are the things that you think we should be researching. And when the community's interests and researcher interests align, the value of that research question, I feel, is exponentially amplified. We all want to be researching things that people care about, and who better to ask than the people affected by the disorders that we're studying. So in summary, we talked about splitting and lumping and trying to do both, transitioning from a linear approach to a simultaneous approach to multidisciplinary science, and then really involving the community in true research question generation and in being active participants in the research process. So thanks, and you're free to reach out to me through the chat or afterwards.
Fantastic. Thank you very much. Our next panelist is Dr. Sarah Jane Webb. Dr. Webb is a professor at the University of Washington and Seattle Children's Research Institute. Her work focuses on investigating the use of EEG and other physiological signals to improve our understanding of individual variability in social attention, cognition, and motivation, and how these systems change over development in children with autism spectrum disorders. Dr. Webb.
Thank you so much. I'm going to get my screen up and sharing. Are you seeing the presenter view or just the
Yeah, presenter mode, sorry.
Do a quick switch of my screens real quick, so we only have one. Thanks you everyone for joining us today. And I want to go in a kind of I really thank Bhuma Avarathan for the points she brought up, and I want to go in a kind of a different direction, and probably because I'm also not a clinician, although I work with clinical populations in the work I do, and I've been thinking about our challenges maybe from a different perspective because of that, and because of roles I play in a number of collaborations, where we really have been thinking a lot about how we acquire data from pediatric populations, and what do we do in that post-processing that really can help us pull out pediatric signals that might be varying in a very specific way based on developmental phenomena that we're trying to address. And so my 3 points that I'd like to bring up as challenges that we're currently thinking about is really, what is resting state and what are resting state experiments? What are they designed for? How do we acquire data in resting state? And what are we doing analytically? And then thinking about reliability, stability, and variability. How do we assess those across our tasks and our, not only in our tasks that we're using, our time points, and as well as our dependent variables. And also how we have a reliance on our field still, I think, on really a reference to a mature or adult model and using that to backward development. And so I'll go into those in a little bit more detail. So here's some images that I like to think about when I was kind of going through these points about resting state and what kind of what I struggle with in resting state, even the work that we do ourselves in this area, whether it be EEG or cardiophysiology or functional MRI, is that we're asking children often to exist in a state that is not normal for them. Children don't potentially rest a lot. If we think about that, they are really active, and more so maybe than we are as adults. And so resting is really, I think, really an active and an explicit task for children in a way that it may not be understood in the way that we think about adults and functioning. And so resting state tasks are often designed to look at intrinsic neural activity that is not elicited through a task, right? And so tasks that are used are eyes open, eyes closed, looking at a fixation point, looking at a viewing a video. And the idea is that this lack of task really allows us to look at wide ranges of populations, right? Because we're not asking the participant to do something. And so we can get this wide range of ages and functioning levels to participate. But at the same time, each of those age groups and each of those groups that we're bringing out in the functioning levels bring something explicit to that task. And I think it can be very different within an age group and across age groups as well as within a population. And so we can think of we were talking about designing a study for kids, sort of in that elementary school to middle school range. And, there'll be some kids in elementary school that have done mindfulness in school. And so when you ask them to rest, they can imagine what it means to sit there with their eyes closed and sort of in a meditative state. And there'll be other children that the idea of closing their eyes is even difficult. We see this, and my own child who is 9 struggles this with actually keeping her eyes closed. And she'll, when she was younger, you'll ask them to close their eyes and they'll have to use something over their hands, and then they'll peek, because they want to be actively and visually engaged in their world. And the idea of closing your eyes and sitting, particularly in a strange research environment, is not something that I think it comes natural to children. And then we think in adolescence and with sleep issues that many of them have, you ask them to rest, and we've seen kids fall asleep within 5 minutes of a task. And definitely when we get into younger adults as well. And then, so we can think about those different ways of that kids choose to rest and how it impacts our variability. And the opposite is that sometimes we give them a task to do, and the nature of that task itself often pulls for cognition, emotion, and processing. Letting a child choose their favorite video to watch is going to pull for a different set of brain activation than letting a child, maybe all children watch the same movie, or doing something very kind of visually basic in terms of thinking about it. So I think we need to think more about resting state and develop common paradigms across it. But also, I think we need to be really explicit about how this interacts. Oops, we're going to get out of this. Sorry. And I think the next one to think about is really defining reliability, stability, and variability in our tasks. And I think this will be some questions we'll come back to. And children have very variable day-to-day activities, right? And so kids are coming in after school or in between nap periods. And I think this is a pretty good example of a comic down in the lower corner where kids are looking at math scores on the day of Halloween, where they're all dressed up and having candy, right? So what's the reliability or math scores on Halloween when assessed on Halloween versus assessed on another kind of educational day? And we have that same variability across participants. And I think across, even when we think about parts of the year in testing children who are coming in and seeing us during the school year are probably in a very different sort of cognitive and learning state than kids that might be coming in during summer break. And I think we all know that the replication issue in our work is very, very much being really investigated and thought deeply about, given that there's many psychological studies. And I think neuroscience and neuroimaging is following this too, where we're really having a hard time replicating studies, not only because of small sample sizes, but because we don't know the reliability, the stability, and the individual variability in our measures, how we acquire them and how we get our dependent variables. And I think sometimes in normative kind of work in neuroscience, we tend to think of like, oh, is this tweet that came out actually last night, or that I thought was really interesting, is where, you know, one of our colleagues says that, you know, what is an extreme outlier and how do we deal with them in our data? Is that a sense of error in our measurement, or is that someone who is not part of the group or not doing the task we're looking at, or, you know, and how do we consider that? And I think when we get into, as Booma says, potentially working with populations that we are expecting more neurodiversity, we are expecting to see more more differences in processing, whether quantitative or qualitative, I think we have to understand really how we address variability and how we include it in our samples. And so I'm guessing that the next slide is also thinking about this reliance on mature models. And so here's some pictures I pulled down to kind of how we think about it. I recently reviewed a study the other day that had infants and adults in it, which I think is a really interesting and maybe unusual comment, because that leaves a lot of development out, right? So if we think about comparing 6-month-olds and 18 to 22-year-olds, I think we have to think very deeply about what we're trying to understand about development or differences or the way the brain does in that very, very large, right, time period. And what are we expecting infants, or are we expecting young children to be just simply small adults, right? That their neural activation patterns should be derived from the variables we are already have articulated maybe in adult work. And I think in the opposite picture, where you can see what we see on maybe memes going around of adults who are reenacting their childhood pictures, right? So here's three young individuals in their plaid pants and their green shirts, and they've reenacted those as adults. And though that's the opposite. So what, you know, you may have may be appropriate in terms of understanding neural activation and functioning in the younger sample when you are at these ages of these kids in elementary or middle school may really not be what is expected to assess in later stages of development up into adulthood. And as anyone can wear plaid pants and green shirts at any time if they want. We're not going to bring down fashion at all, but I think it shows that I'm guessing this probably wasn't what their normal clothing was or choices that they made. They probably don't all dress alike anymore. So I think the idea that how we really think about understanding change over time, both within an individual and in cross-sectional samples, is really important. And this gets back to our individual variability question as well. We see this picture from Sound of Music, where you can see everyone's dressed in the same clothes, right? But that same task or those same outfits really fit everyone in this picture differently because of their age and because of other characteristics. And so understanding, again, as that individual variability, how we look for common signal, but also how we keep in mind how development is both, I think, what is maybe kind of a universal or a common process, but also might have a really strong degree of variability in it in order to have a learning system that's moving forward and maturing. So I'm going to leave it at those 3 comments, and I look forward to hearing what Dr. Pomeroy is going to bring up.
Fantastic. Thank you very much. So our next panelist is Dr. Scott Pomeroy, Dr. Pomeroy is the Bronson Crothers Professor of Neurology at Harvard Medical School and Chair of the Department of Neurology at Boston Children's Hospital. His lab focuses on embryonal brain tumors. He has served on many NIH study sections and currently serves as co-editor of Neurology and Clinical Practice and associate editor of Annals of Neurology, as well as president of the Child Neurology Foundation. He also is an elected member of the U.S. National Academy of Medicine. Dr. Pomeroy.
Thanks, Adam, and Sarah and Booma for helping set the stage here. So really at a really exciting time in child neurology and in developmental neurosciences more broadly, where we now have a very sophisticated or growingly sophisticated understanding of how the nervous system works. And we have a fairly sophisticated notion of the genetics underlying neural development and the disorders of neurodevelopment. So how do we implement this knowledge in the most effective way as we go forward? As we come to understand diseases at a molecular level, we really understand that there's quite a substantial genetic variation, and there may be allele-specific symptoms or even allele-specific treatments that we have to implement, which kind of gives us the scary notion that every individual is going to need their own specific treatment. We kind of worry about this in cancer, as each cancer is so sufficiently different genetically that we're going to have to have a specific treatment for each cancer as we're giving drugs that target mechanisms. So what we really need to do is try to find final common pathways that we can understand at a sophisticated level so that we can identify targets of intervention that will apply to more than just one individual. And it's one of the big challenges we face both in cancer and in neuroscience. And to do this, given the heterogeneity of what we have, we need to be able to see the big picture and then to be able to work collaboratively to find ways to define disorders in such a way as we identify these final common pathways. As Booma pointed out, how do we lump in a sophisticated way that's driven and informed by a mechanistic understanding? The challenge is further complex by the dynamic nature of the developing brain and indeed the dynamic nature of a 3-year-old who's awake and running around, and you're trying to gather information from kids, as Sarah has pointed out. So how do we define meaningful and reliable outcomes that are context defined, context in terms of the environment they're in, but also the context of the developing brain? So I think these challenges kind of provoke us to think of several issues that we now need to address, and which is one, how do we define diseases in such a way that we, using preclinical data, can define mechanisms that then we can convince pharma or others to develop drugs to target. So biotech, pharma, at the end of the day, we're going to need new treatments to treat these conditions that we understand at a mechanistic level, unless we're so fortunate to have existing treatments that can be repurposed for that. So that what type of data is needed, what fidelity, what quality, what are the data that are needed to drive industry to develop new therapies? It also is important to us that we work collaboratively to define very high fidelity data that are both reliable and reproducible, and that we have outcomes that are measurable and quantitative. So if we're treating a condition, we can be reasonably sure in a relatively short period of time that the treatment either is or isn't working. A challenge that we must meet only meet if we work in a way that challenge ourselves to develop outcomes that are statistically robust. And finally, I think as a field, we need to teach ourselves how to run investigator-driven clinical trials. We're pretty good at running industry-driven clinical trials. But I don't think, having lived in the world of oncology now for 3 decades and the level of sophistication of clinical trials in that space, I think we have a lot to learn within child neurology and the developmental neurosciences, how we can make sophisticated clinical trials that meet the challenges that we're faced with, given the genetic heterogeneity, the dynamic nature of the developing brain, and the dynamic nature of the things that we're trying to measure, and do it in a way that we can bring the greatest good to our patients and their families as we develop new therapies in this modern era. So I'll end there and look forward to further questions coming our way.
Fantastic. Well, I wanted to thank all of our panelists, and if they all put their cameras on. And so we're going to start the question and answer period now. We have a series of questions, so I'd like to ask the presenters to keep their answers informative but brief. And as a reminder, if you want to ask a question, you can click on the Q&A section on the lower right part of your screen. So let me start with a question that I guess, we've posed these challenges, and what we'd like to hear is some potential solutions for all of these. And so I'd like to give everyone a chance. We'll start with Buma. So one of the questions that came out was splitting and lumping is great, but traditionally reviewers struggle with this concept. So how can we address that?
Yeah, it's a great question. I think that lumping has to be done in what I tried to describe as an intelligent way. So I think part of the problem with how reviewers have addressed lumping, I think, is a fair one, in that you can't, for example, consider all people with CP as one homogeneous group. And I think when you say you're studying a specific population, you could pull out certain characteristics of that population and justify why they should be cohorted in a certain way. So maybe you only study for this particular mechanism, or if you're interested in this particular type of circuit dysfunction, say I'm only studying people who had HIE, born at term, who were cooled, who are manifesting this type of motor dysfunction at this level of functionality, gross motor functionality. And it is a starting point. It's not ideal. And I think acknowledging that up front and saying that you're doing your best and that these are the criteria you're using to narrow down your cohort, in my experience, has been viewed favorably by reviewers. But starting out saying, it's heterogeneous, what am I going to do, is a different bag altogether. So I think being honest, but also saying that you're doing your best and here's what I'm putting out there, is valuable.
Did you want to propose any solutions for your other challenges?
Oh, yeah, sure. So I talked about the second challenge, going from linear to simultaneous. I really do think that's a funding problem. I think when you put out a grant saying you're trying to do all of these things at the same time, I think it can often be criticized as overambitious, and something like this, how could you possibly ever get it off the ground? So I think there should be funding mechanisms created for specific problems that fund groups of researchers who are willing to come together to address these aspects of an issue simultaneously. And there are some private foundations that are interested in doing that and are developing ways to do that. And I would love to see traditional funding organizations do that as well. I think if you put the money out there, people will come. And I really do think that's the way to get the second thing off the ground. The idea of addressing all aspects of a translational scientific project simultaneously. The last thing I talked about, involving the community, that's the easiest one, I think. The community is eager to get involved, and they need two things, I think, to address the barriers to getting involved. One is, I think there is an intimidation factor. I think if you ask someone what their thoughts are about the research in the specific disorder that is affecting their child or is affecting them, there are some people that feel like, who am I to contribute to this? So one aspect of that is education. And I think we're all getting better at doing that, putting out webinars and content that is specifically targeted to the community. And I think we should all do a better job of that whenever we publish anything too. Putting out infographics, we see a lot of these on Twitter that are meant for the lay public. Making sure to share your research with community organizations as often as possible. And then in addition to that education, it's creating a platform for people to participate in research setting agendas. So interestingly enough, there's Laura Gilbert, who's a resident at WashU, will be speaking about many of these issues at an upcoming conference in Child Neurology Society conference, about creating a new platform for the community to participate as well. These platforms exist, the community is energized. We just need to open the door. So I think that the third one is the easiest one to solve. I think we just need to focus on it.
Thanks. So let me ask, Sarah, did you have any comments on Buma's challenges?
Yeah, I think her second one about funding is a really interesting one, and we can always say we need more money, right? And what the mechanisms are. But I think what you bring up is potentially, so there are currently mechanisms for center grants, right? And that type of thing, but they tend to lead towards more experienced, established investigators with R01 size projects, right? And I think the idea to really kind of think how we link projects together, sort of like linked R21s, which are high impact, right? High risk, high reward, or grants that call for a mix of those to be together into a package or something like that, I think could get at what you're saying, because often some of that work around doing it simultaneously, right, is not currently in our thinking, and it is going to take actually some failures, right? It's going to work, or it's going to be high risk, and it's going to be, it potentially has the possibility of failing if you miss. And so I think that those kind of ability to, or there's the there's a, is it R34s, R61s, right, where you initiate, then you get feedback and you go to the second grant. Those types of more grants that have a pause to come back and reset after the first 2 years, wherein aims 1 and aim 2 are set because we know that these are established, but aim 3 is going to be really impacted by what you do. And I think sometimes those are hard to get funded, and maybe that impacts what you're seeing and people sort of seeing, and potentially things like lumping and splitting even, like we're going to try this, but it might fail. And that's where we get into review problems. Like, oh, I don't think that's the right way for you to lump or to split. Wherein, what if we tried it, and because you think the idea of lumping and splitting is great, we're going to come back and show that it works, and then that's going to inform the next day. So I think there's some ways we can creatively think about how we bring in levels of research in there and do it. I think the other ones are, really great points about community engagement and lots of groups, and particularly in the field I work in, autism, there's a really strong movement in making sure that the individuals that affected by the disorder or are most involved in it. I think it's an interesting question for broader pediatrics. When we're talking about big population issues, there's a new research funding announcement out, on media use, right, which is going to hit all kids, right? And so there isn't a parent group that you go to be like, parents sometimes really care about media use, and they sit on this whole wide spectrum. And so how do you involve the community in planning grants like that? So I think there's the rare disease model, right? It's people who have very specific and labeled conditions. And then there's those that sit on that really spectrum area where they're maybe have multiple diagnoses or are not diagnosed for a number of really significant medical, because there's barriers to diagnosis. And then there's this big public health one. So I think it's interesting to think about what does community engagement look like across all of the levels.
Thanks. Scott, your thoughts on Buma's challenges?
So I'm going to fall back on my familiar territory and tell a story about cancer, because that's what I've done. And in medulloblastoma, we've known for a long time that even though they all look the same under the microscope, there are some that respond to treatments and others that don't. And so we're not informed. We can't tell by how they look under the microscope the difference between one versus the other. So many of us throughout the world can put together our samples into large cohorts and use sophisticated molecular analysis. And we discovered that they're actually break down into very discrete subtypes. And there's one, so we can take this and lump within this a group of tumors that are all driven by, about a third of them are driven by the sonic hedgehog pathway. And so you have to say, well, great, lumping has really given us deep insight at a molecular level of what's going on there. It's the sonic hedgehog pathway. So let's block the sonic hedgehog pathway. Well, it turns out that the current treatments for the sonic hedgehog pathway tend to inhibit a molecule called smoothen, which is rather proximal, and it's at the cell membrane level. So all the internal signaling mechanisms beyond that are downstream of where the drugs are effective. So now we need to split again. There are those mutations that are upstream of that point that are absolutely blocked by the inhibitors. They're very effective. But then those activating mutations downstream of smoothened are completely insensitive to that treatment. So now we need to split again. So I think the way I see it is what we need, the lumping and splitting needs to be done at a level that we can understand, that it's an informed, a mechanistic. If we're going to use targeted things to go after, we need to understand the mechanisms we're targeting in the context in which they're activated. Now, let's jump over to autism. We sequenced all these patients with autism, and by golly, we found like there are 1,000 autism genes, and nothing really stands out, and at a genetic level, or very little that tells us anything that's useful. You couldn't do anything with that information of value in terms of a treatment. One of the impulses of this is we need to increase the N. We need to get a bigger sample. Well, what you're going to do if you do that is, because of the heterogeneity of the disorder, is you're not going to clarify anything, I don't think. So what I believe we need to do is to get really smart, sophisticated people to start looking at things that start lumping in various meaningful and mechanistic ways, maybe by symptom, maybe by a brain structure. I'm not sure what the right answer is, but my guess is you'll improve the signal to noise ratio, and you'll maybe start to find some genetic signals that pop out if you can then cut down the amount of heterogeneity in the sample that you're looking at. So, and then you may need to split again, but do things in a way that it make it so that you have the greatest chance of a signal to noise ratio that will give you a clear either genetic or other outcome as you're moving forward.
That's great. Thanks. All right, Sarah, you're up. How are you going to address your challenges? You can pick one if you want to. I'll have the others comment on, or you can go through all of them. It's up to you.
I think that one I think is goes back to kind of Booma's comment about what we ask for in research studies and what we ask for in publications. So this is like where we come top down, like you said, for funding. But I think this is the second one about defining reliability, stability, and variability, is that I think that we really have to move towards requiring replication samples or reliability metrics and more psychometrics in the work from the beginning that we do, and really considering that to be a part of the methodological process of any grant that's going out there, and part of the first types of publications that come out of the work that you're doing, right? So we think pilot studies of like, does this work, and can I get the kids to do it? Can I acquire the data, and do I get a signal out somewhere to show, and then acquiring data. But I think that we really have to move towards kind of some people, it's boring, right? In some ways, is to to make somebody come back a day and do the same thing again, right? It's not, we get nicked sometimes, I think, for not being innovative, because we say either I'm going to do the same experiment I did before, or I'm going to, or we're going to do it twice, and then not having that data ahead of time, can impact it. But I think we need to start making that parts of aims and parts of methods that are just what we do moving forward. And I think that these issues about how we define reliability, whether it be split half or whether it be day to day or a week later, or, you know, but depending on the age, based on what we want to use the signal for, right? And so I know, Adam, we've had these conversations about biomarker use for clinical trials, right? And thinking about what, if we want to show that a biomarker predicts treatment outcome or is going to predict some type of change in clinical symptoms, knowing how stable it is either in the moment of acquisition, the first half of the trial, so the second half, or a day later. So, you know, think about clinical trials that might have a baseline and then a first dose response, or 6 weeks out because that's the second time point of, you know, of a trial. I think we need more studies that incorporate that, even when doing other aspects of neuroscience that aren't quite ready for clinical trials yet. So I think this aspect of psychometrics and statistical, maybe this gets into, you know, a little bit what Scott says, as statistical knowledge and innovation in how we do both the basic really good stuff to make sure that our neuroscientific methods are the same, are considered in the same way that we might develop an IQ test, right? That we need, if we're using IQ for good or bad in some ways, or a Vineland or something to tell us about clinical prognostic or developmental prognosis, and we want to insert neuroscientific methods in there, we want to insert a biomarker in there, then we have to be held to the same standards, right? And so this conversation about what is reliability for these measures that are going to have different types of signal and noise, I think is one we really need to integrate more fully into the work we do up front. And I think that handles some of the other problems, right? Because questions are like, what is resting state? Well, we know, you know, if you're doing it twice or you're doing it under these metrics or you're sharing your experiments, I think that's some of the great things about some of the large consortiums where we have been, our experiments are now open source basically, right? So we're sharing, you know, in the Autism Biomarkers Consortium, we're sharing paradigms with the EU AIMS LEAP program, ABCD, and all these letters, right? In terms of the neuroimaging consortiums, those, you know, ways of acquiring data are now being utilized in other studies, as we can see people say, oh, I'll pick up this ABCD scan and then I'll run my new stuff, right? And I think that really gives you really a better and deeper understanding, because you have this ability, maybe in Scott's ways, to compare to other samples, to compare to, you know, the sample you couldn't enroll, right, from that is published by somebody else, that gives you that big N, but at the same time, you know, being able to focus on novel ideas at the same time. So I think those kind of methodological questions will move us, methodological focus, as well as some of the big picture issues, will help us really be able to solve some of these questions, or at least address them. We may find that our measures are not reliable, and then we're going to be back having a different challenge conversation, right, Adam?
That's great. Scott, your thoughts on any of Sarah's challenges?
I think in addition to reliability, the focus on sensitivity and specificity is important. So, you know, IQ is pretty reliable, but it's not very sensitive. You know, you'd have to have a pretty massive effect to move an IQ point. Whereas if what you're really interested in is kind of a neural mechanism that's related, maybe a visually evoked response, looking at a quantitative number that if it moves a little bit, it's pretty reliable, but it's also sensitive and fairly specific. And that may be a starting point. It may not be the final point that you want, but at least you get a signal that you can rely on that says, okay, I'm moving in the right direction. Let's keep going. I think there's a tendency in cancer, you know, to swing for the fences. I'm gonna, you know, dunk a thing into a dish of cells, and then I'm going to give it to a child, and maybe it'll cure the cancer, you know, and you'll get lucky. That doesn't work too well, I have to say. You know, how many failed clinical trials are there? But as we get more and more sophisticated and say, okay, what are we looking? What's a signal that's reliable? That's really logical and makes sense, moves us in the right direction, and then go. You know, don't just swing for the fences. Understand what you're going for, and I think you'll get. My belief is you'll get there sooner than the people who swing for fences.
I think that's a really good point, Scott. We've had this conversation about, well, if something's too reliable, it doesn't change, then why, you know, is that something that you're going to actually try to treat? So we need some signals that they have movement in them, but also have the right psychometric properties to know that we're really assessing what we think we do.
It's that the trick is making sure you're not just studying something that's meaningless. I totally agree with that. But on the other hand, you like it to have sensitivity to what you're trying to do.
That's great. Booma, your thoughts on Sarah's challenges?
I definitely agree with the kids are not little adults aspect of those comments. I think very much true, and in a lot of ways, I think we've tried to fit, you know, square models into round kid-sized holes for a lot of studies for a long time. And I think transitioning to really focusing on pediatric models for pediatric disease is valuable. To piggyback on I know we're talking a lot about reliability, but obviously it's critical. I think that there is a lot of value in being internally consistent in your own lab. And I know setting up my lab now, I'm repeating a set of experiments that I've already done because I'm in a new space in a new room, and I want to make sure that all the results are comparable. But I also, and obviously that takes time, and that takes money, and that takes effort. And it's valuable because the science requires it. But I wonder, you know, is there a way to get other people to replicate some results I'm putting out there and be rewarded for it so that they want to do it too? And I think that's been difficult because, you know, you're not going to get your results published if your results are, if you know, it takes a year for you to replicate someone else's results, even though it might be critical for you to do that in order to get to the next step of your research if you want to build on what someone else has done. So I think particularly for young investigators, it may be helpful to have a mechanism, particularly in larger journals, where replication of results can serve as replication of large papers can serve as almost addendum publications to the original publication. I know this is kind of wild and out there, but in a world where currency, you know, in a publish or perish scientific world, if you're putting the work out there to replicate others' results and you're hitting gaps or hitting problems, and it's expected that when you put a paper out there, other groups are going to try to replicate your results and their papers are going to get attached to yours. It's in the interest of the parent lab of those results to help you replicate their results. And it's in your interest to replicate them because you get an attachment paper to a big paper that's already been published. And I don't know if this model has been tried before, but I do think there needs to be some sort of tangible, something tangible to show for other labs investing in replicating a different lab's results.
That's great. I'm actually scratching a bunch of notes down as you guys are talking. That's an interesting idea. Scott, so we only have a few minutes left. Scott, did you want to offer any potential solutions to some of the challenges that you brought up? Maybe because I want to make sure we have enough time for Booma and Sarah to comment too.
I think that, you know, increasingly today, team science is what we're all about. That to bring, as Booma says, communities together is really key, and so that we can work together and get large enough cohorts and work together so that we can start to understand at a pretty sophisticated level the heterogeneity within them, and that's meaningful, and hopefully can split into meaningful subtypes. I think the challenge then is, okay, well, how do we, in a competitive world where everybody, you know, wants to move forward, how do we float all the boats? How does everybody move forward in an academic setting, and how do you remain competitive for grants? And I think to remain competitive for grants, it's going to be based on the quality of your work. If you are part of producing very high quality work that is clearly moving in the right direction, you will get the grants. I'm pretty sure, you know, or learn how to write grants better. But your chances of it's a lot easier to write a grant if you have compelling data and a compelling story that you can push forward than it is you're working on squishy data and you're really, you know, you're not sure where it's going to go. So I think one, high quality data, work together, work with people who are going to share with you the glory, and look out for one another's interests, and establish your niche and move forward. I believe that's a way forward, and for at least that has been in the field that I've been in.
That's great. Booma, thoughts on Scott's challenges?
Yeah, I think the team science approach is critical, and I think I vaguely echoed some of that in some of my points as well. I think it's I would say a barrier to maybe cross to kind of facilitate team science is this, and this may seem tiny, but we see more and more papers being published with 3 co-first, 4 co-last. And I think that's important because it shows how many people are required to push projects forward. I think that idea of author order and equal contribution is viewed still in a very traditional way when you're thinking about reviewing what that paper means in terms of contribution to a person's career. So there is such a thing as being third co-first is not as good as being first co-first, and being penultimate co-last is different from being last co-last. And that requires a bit of a culture shift, I think, on the part of grant reviewers in particular, to know that last is last, equal contribution is equal contribution, co-corresponding is co-corresponding. So it's a small change, but it's a big culture shift. And I think that does need to happen in order for team science to happen without that asterisk. So I think that'll be, and we do see it happening, but it would be nice if it happened a little bit better.
Great. Sarah, you get the last word.
Yeah, well, I think that Booma brings out something that it sits on, your shoulders, Adam, and mine and Scott's, for being senior people in the field, that when we review somebody's packet, what we should be asking is, what's your individual contribution and what's your team science contribution? And that's also how you get it, is you change. We, as people who make those decisions in promotion and those type of things, need to be the culture change one, so that we acknowledge that the younger generation, new faculty, are doing really fantastic team science, and they should get all the credit for it.
Great. Well, this has been a really fantastic discussion. I really wanted to thank all of our presenters and our attendees. So just as a reminder, the session was recorded and it will be transcribed and then archived on the NINDS website in the coming week or 2. Details about where to find the archive version will be posted on social media and professional society listservs and newsletters. And obviously, if you have any questions, you can always contact me via email. So I hope everyone has a really great weekend. Thanks a million for your thoughtful contributions to both the presenters and the audience. And we will see you until next time in our Challenges in Pediatric Neuroscience Research webinar series. Thanks, everyone. Appreciate it.
Thank you.
Thank you.